Hello everyone, my name is Jessica Tolbert. I am a student at Claflin University. I had the pleasure of working at Hunter College under the Leadership Alliance program with Dr. Ortiz and lab. The project that I worked on was identifying factors involved in barrier insulation within the mouse T cell receptor alpha locus control region. So first I'd like to begin by establishing what I'm actually going to be talking about. So here you have every cell, and every cell contains the same genome, and every cell has like its specific job and a specific purpose. So, and the genome is made of DNA that can express different genes. So here I have eyes and a hand, and basically I'm showing that because every cell contains the same genome of DNA, what allows, say, the eye to not express the genes of the hand, and the genes of the hand to not be expressed in the eye, and vice versa, and that type of thing. So what allows this to occur? What allows this gene regulation? So differential gene expression is controlled by DNA regulatory elements. So a type of DNA regulatory element is an insulator. And insulators basically just insulate the gene. They allow genes to be expressed without the influence of other genes within the genome. So here we have two types of insulators. You have your insulator blocking insulator, and then you have your enhancer blocking, and then chromatin barrier. Your chromatin barrier is shown here in the diagram, and it can block closed chromatin from closing open chromatin. And here you have your orange circles, which are the closed DNA, and it's tightly compacted around, and the DNA is tightly compacted around histones or proteins. And then you have your open DNA, or open chromatin, which are the little green balls. And the DNA is more open here. You can see the higher marks. So locus control region, LCR, provides integration site independence. And LCR, or that I'll refer to later on in the presentation, is comprised of DNA hypersensitive sites, which are also, you can think of them as a subunit or subgroup of an LCR, and they're kind of like bricks. They're the building blocks of the LCR. So there's three functions of the LCR, the first being that they allow transgenes to be expressed no matter where they're inserted in the DNA. And you also have LCRs within their natural location to be able to perform their job over long distances. And then the last one, they facilitate expression in the right place at the right time. So here I have an example of gene A that's linked to LCRA, and it's expressed in the eyes. And so then if you wanted to insert gene B where gene A is, you would link it to LCRA, and that particular gene B will be expressed at the same place at the same time in the eye that A would be expressed. So the LCR that we're interested in, or the locus control region that we're studying in my lab, is the T cell receptor alpha locus control region, or that I'll refer to later is the TCR alpha LCR. And this particular LCR is made of has two genes linked to it, DAD1, which is widely expressed in many tissues, and TCR alpha, which is only expressed in T cells. Now, the reason that this LCR is of interest to us is because it allows two differently acting or differently expressing genes to be expressed no matter where. It allows it to be expressed wherever it's supposed to be expressed, despite their activity. So it led to the question of, does this LCR have insulator activity or within it? So studies were done by Ortiz and Harrow, which basically so basically all they did was they took this particular LCR of interest and they looked at one specific subunit, which is hypersensitive site 6, or HS6, as you see here. And they basically saw that there was activity here that allowed for this particular LCR to act as a barrier insulator, which I mentioned earlier, allows the closed chromatin to not close the open chromatin, or the expressing genes. So then further research was done within the lab, and it showed that this particular LCR, again, has barrier capacity due to a 90-day essay and other research. So back to the title, which was identifying the factors that relate to the barrier capacity within the LCR. And so the candidates or the factors that are noted here are ELF1, AML1, or also known as RUNX1, and RNA polymerase II. AML1 and ELF1 were found within the lab, but RNA polymerase II was found on an online database where RNA polymerase II was shown to be attaching to our specific LCR. Now, this is a very interesting thing because RNA polymerase II is known to produce transcripts during DNA replication. So, and it usually attaches to the promoter of the DNA. So this chromatin barrier insulator here, or this particular DNA regulatory element is not a promoter. So it's very interesting that RNA polymerase II is even attaching here, and we're trying to understand how it's actually, what its role is in the barrier activity of our specific LCR. So here you see it's attaching to hypersensitive site 6, or the subgroup that I mentioned earlier that we're looking specifically at. So like I mentioned earlier, it normally produces transcripts, but here, we're thinking that maybe it's recruiting other enzymes that allow this particular chromatin barrier insulator to continue on with this function. So this leads to our hypothesis of RNA polymerase II binds to hypersensitive site 6 and could play a role in barrier activity, as I mentioned earlier. So in order to understand what it's doing and whether or not it has anything to do with this particular barrier activity, first we worked with, we identified a cell line that we could work with, which was the VL3s or T cells. And then from there, we identified inhibitors that would prevent RNA polymerase II from phosphorylating, because the particular RNA polymerase II that we're looking at is phosphorylated. So if you prevent the phosphorylation, then you prevent the activity of that particular RNA polymerase II. So THZ1 and BS-181 were the two inhibitors that we used in our experiment. In order to understand the amount of inhibitor we needed to use, we conducted the MTT assay, cell viability test. And this cell viability test will help us identify whether or not the cells are viable, or how much inhibitor we use so we don't actually kill all the cells. And then from there, we would identify the proteins by conducting a Western blot. So here's the optimization of the particular inhibitor. So we conducted the MTT assay, and there were several variables that we had to change in order to make sure we weren't killing the cells with the inhibitor. So first we had to actually see how much, we had to change the amount of inhibitor dosage and the number of cells and the incubation time in which the inhibitor was allowed to incubate with the cells over a period of time. And here I just included a 90-well plate that we used in order to look at the MTT assay. So here, the first chart at the top left, you'll see that we had to actually see how many cells we needed in order to be able to see to have a good marker. So we ended up using 0.75 million cells, and from there we saw that the cell viability using the THZ1 chemical inhibitor, the cells died after 100 nanomolars of adding the THZ1. So the range of 1 to 100 concentration is what we'll be using later. And then for the BS-181 chemical inhibitor, we saw that the concentration needed without killing the cells was between 1,000 and 10,000 nanomolars. So here I wanted to show the importance of the Western blot to my particular experiment and what we hope to see later on. So here you're adding the different antibodies to the protein, and these particular antibodies will identify the protein in question, which is RNA polymerase II. And from here you will understand how much RNA polymerase II is within your particular cell. So after it goes through those different processes, then you will have your amplified protein, which will allow you to see how much RNA polymerase II is still there. Future goals would be to identify the impact of RNA polymerase II inhibition on the T cell or alpha LCR insular activity by continuing to optimize conditions for the Western blot and then narrowing down the concentrations that inhibit RNA polymerase II within the established range. Future implications of our findings. So the basic science implication would be to provide new directions for investigating LCR activity. And the applied science implication would be diversifying the pool of potential insulators that could be used during gene therapy. So I would like to thank the Ortiz Lab, Dr. Benjamin Ortiz, Guy 3, Jordana, Zed, Leadership Alliance, and the Hunter College SPUR program, as well as the first year research experience program through the Leadership Alliance. Thank you.